Good morning. You can all start the show. Kalpana, you can start.
We can start, ma'am.
Yes, you can start. Yes, you can start.
Respected guest, head of the department, faculties, and my dear students. A very good morning, one and all. I am Dr. Kalpana VN, Assistant Professor from the Department of Microbiology. I welcome you all on behalf of the department to the virtual seminar Multidrug Resistant Bacteria, a significant threat to global public health. Let's begin the session with prayer. Our Father, who art in heaven, hallowed be thy name. Thy kingdom come, thy will be done on earth as it is in heaven. Give us this day our daily bread, and forgive us our trespasses, as we forgive those who trespass against us. And lead us not into temptation, but deliver us from evil. Amen. Now, I would like to request Dr. R. Manjula, Assistant Professor from the Department of Microbiology, to introduce our resource person and welcome the gathering. Over to you, ma'am.
Good morning, everyone. I am Dr. R. Manjula, and it's my privilege and great pleasure on the behalf of my entire department welcoming Dr. Udaya Lavanya Ma'am, Assistant Professor, Department of Biomedical Sciences, Sri Ramachandra Institute of Higher Education and Research, Poru, Chennai, for taking the precious times out and gracing the occasion. I would like to extend my gratitude towards our head of the department, Ms. B.V. Gauri ma'am, for being our pillar of support, encouraging us in all the aspects. I feel honored to welcome our department faculties for taking part in this significant ritual seminar. Last but not the least, I take pleasure in welcoming our dear students, the young minds, the future researcher and scientists for this ritual seminar on the topic multi-drug resistant bacteria, a significant threat to global public health. To introduce Dr. Udaya Lavanya ma'am to you all, I would like to list down few of her achievements. Ma'am has done her masters in biomedical genetics at VIT Vellore. Ma'am completed her PhD on the topic entitled Anti-tubercular Drug Induced Hepatotoxicity in Rats at VIT Vellore. Ma'am started her career as assistant professor. Currently, 2.5 years of work experience. Ma'am published around 31 papers in various journals, national and international journals, with cumulative impact factor of around 21 plus. Currently, she is working on project related to drug-induced hepatotoxicity in human liver cancer cell line. It is indeed our honor to have such a pleasant and dynamic personality whose presence will surely inspire and encourage all of us, especially our students, toward the field of research. A hearty welcome, Dr. Udayan Ravanya ma'am. I could assure you all that we all would feel enriched and raised in the levels of knowledge to a newer height. Once again, I welcome you all and hope all would have a great time ahead. Thank you.
Thank you, ma'am, for your warm welcome. The main objective of this virtual seminar is to increase the awareness of global antimicrobial resistance, encourage best practices among the students, the general public, health workers, and policy makers to avoid the further emergence and spread of drug resistant infections. Taking this idea forward, I request our resource person, Dr. Udaya Lavanya ma'am, to start today's seminar. Over to you, ma'am.
Thank you, Dr. Kalpana and Dr. Manjula, for your warm welcome. And I am indebted to thank our patron, Sister Alice, the convener, Sister Jay Shanti, and the co-conveners and organizer of this event. And I'm thankful to Gauri ma'am, the head of the Department of Microbiology, for giving me this opportunity. I take immense pleasure in doing this talk for you, and I am extremely honored. And I take this opportunity to bring about awareness among students about the use of antibiotics and how harmful they could be to the society if taken inappropriately. And I also thank the momentous advisors and the faculty coordinator, Dr. V.N. Kalpana, for making this possible. And I think we can start off with the seminar. Could you please project the slide, ma'am?
Yes, ma'am, I'll do it.
Thank you, ma'am.
Can you see the screen, ma'am?
Yes, ma'am.
You can go about. Thank you, ma'am.
Thank you, ma'am. So the topic for today's seminar, as you know, is multidrug resistant bacteria, a significant threat to global public health. And to go on about the introduction to this particular interesting topic and a very responsible, thought provoking process this seminar could be. So, to have a few points as introduction for that, we all know that antibiotics are drugs which are used to treat infections which appear due to microorganisms. So, the general term antimicrobials would address those agents which are used to treat microorganism infections, like microbial infections, like which are of bacterial source, viral source, and those caused by fungi, and also parasites. Specifically, micro the malarial parasites, they're very much known for developing resistance. So today we'll be much focusing on antibiotics, and we know that the first antibiotic that was discovered was penicillin, and the scientist who discovered that was Alexander Fleming, and this happened in 1928. And since then, we know that there are several other antibiotics which were discovered in the subsequent years. Till today, we have many antibiotics coming up, but the appearance of newer antibiotics, very much newer antibiotics in the market, is slowly losing its space, because we need so much of research and development and investment in that area. So the idea that any infection could be cured by the use of antibiotics is not an appropriate thing. So we should not think that immediately an infection shows up, you have to take antibiotics. So that's not the way it has to be approached. And apart from that, over-the-counter medications, approaching medical shops and getting medications on our own are also a main reason for the global increase in this kind of threat. Okay, so the antibiotic consumption globally has increased drastically over the years, and today almost about 100,000 tons of antibiotics are manufactured every year globally. So this keeps increasing every year. And the use of antibiotics, it has made a profound impact on the life of bacteria in the world. And we know that bacteria, like any other organisms, they evolve over a period of time. They tend to evolve. So this evolution happens for the survival of the bacteria, like how we tend to use different tactics and strategies for our survival. Likewise, the bacteria and viruses and fungi and the parasites, they also tend to use certain strategies for their survival and existence in the world. So the use of antibiotics is actually to target these microbials, but instead they tend to come up with different kinds of strategies to cope up and survive the attack of these antibiotics. So that is what has been happening throughout these years, and bacteria, they've developed significant amount of resistance to the commonly used antibiotics, because when we tend to take antibiotics for granted, like improperly taking the dosages and taking unnecessary antibiotic dosages, so all these have mounted to this kind of an impact, where the bacteria have learned over these years like how they could survive the attack of these antibiotics. Next slide, please, ma'am. Yeah. Now about antimicrobial resistance, we know that these microbes, they have to survive and grow by resisting the action of antimicrobials when we take in these agents. So it takes a lot of time for these bacteria to develop different kinds of resistance mechanisms. So there are specifically known clinical strains which have adopted specific mechanisms to develop specific kind of resistance to combat the attack by the antimicrobial agents. So microbes, they gain resistance to antibiotics specifically by particular mechanisms. I have listed around 6 mechanisms, and I'll be giving you an explanation about 3 with examples. Okay, so the first one is mutational alteration of the target protein. So any antibiotic will have its target, and the bacteria are very smart enough to undergo mutation of these target proteins to escape the attack by the antibiotics. And the second category is acquisition of genes from other species. Like for example, the E. coli acquiring genes for resistance from some other species. Okay, so that would modify the target protein present in E. coli. Okay, and the third one is enzymatic inactivation of the drug, which is actually a biochemical resistance mechanism. And the fourth is target bypass. It just doesn't reach the target. The drug doesn't reach the target. So the access to the drug could be prevented. And the last one is multi-drug efflux pumps. So some of the bacteria tend to have efflux pumps for the antibiotics, so that the antibiotics actually don't tend to do their role. Next slide, please, ma'am. Yeah. So this picture gives an idea of what could be the antibiotic targets and how antibiotic resistance could be. So here you can see that antibiotic targets are of different types. Like cell wall could be a target for antibiotics like beta-lactams and vancomycin. Likewise, DNA or RNA synthesis, that could be the target for specific antibiotics. So here the examples are fluoroquinolones, and these are synthetic ones, and then folate synthesis. Folate is also very much essential for the function of the cell, and folate synthesis could be a target for specific types of antibiotics. Examples are the sulfonamides. And then cell membrane could be the target, and daptomycin is one example of an antibiotic which actually acts on the cell membrane to kill the bacteria. And then there are antibiotic targets which are protein synthesis. Okay, so these targets are targeted by antibiotics such as linezolid, then tetracyclines, the macrolides, aminoglycosides. So these all are examples of antibiotics which target protein synthesis and thereby prevent the survival of the bacteria. So now how these antibiotic targets escape the attack by these antimicrobial agents. So now they develop antibiotic resistance. So like we listed in the previous slide, the efflux pumps, they are one of the major resistance mechanisms. And then the second could be immunity and bypass mechanism, and then target modification, like I told you, mutation of the target protein or alteration of it, and inactivation of enzymes. So this is a most popular one and a very challenging one in the clinical side. So the beta lactams could be attacked by the enzymes called beta lactamases. Okay, so which can inactivate the beta lactam drugs can inactivate these enzymes. So other examples for the same category of inactivation of enzymes is aminoglycosides, which are very commonly used. Next slide, please, ma'am. Now, the first one that we're going to look into is mutational alteration of the protein. So I told that fluoroquinolones are man-made compounds. So it's very unlikely for these compounds to be inactivated by enzymatic mechanisms. They can escape that kind of resistance mechanism, but still they can become resistant by acquiring mutations. So mutations can make the target protein of the bacteria very less susceptible to the antimicrobial agent. So they become very much, they tend to resist the action of fluoroquinolones by acquiring mutations in the target protein. So this kind of fluoroquinolone resistance is mainly due to mutations in the target enzymes, and example is DNA topoisomerases in this case. This is not very exclusive for fluoroquinolones, but this is one of the major mechanisms that is being followed. Next slide, please, ma'am. Yeah. The second category of resistance mechanism is acquisition of genes. So these resistant genes do not actually belong to that particular bacterial strain, but instead they are acquired from some other species or some other strain. So an extreme case of this scenario is the generation of MRSA. So we all know that MRSA is nothing but methicillin resistant Staphylococcus aureus. You know that Staphylococcus aureus is a normal flora that is present on the skin. So MRSA strains, they contain a new methicillin resistant PBP. So this is called PBP 2A or 2-. So the expression of this particular PBP is often induced by drugs like methicillin or other beta-lactam category of antibiotics. So the gene for this one, it is located in a large segment of DNA, but still we don't know how this large segment got transferred to Staphylococcus aureus from another organism. So it is not originally that of Staphylococcus aureus. Rather, it was transferred from another organism into the Staphylococcus aureus genome. So we know that plasmids are very much capable for transfer of genes. So acquisition of genes is one important mechanism that bacteria adopt for developing antimicrobial resistance. Next slide, please, ma'am. The last example that we are going to see is that of enzymatic inactivation of drug. So there is common resistance mechanism for antibiotics which are of natural origin, not man-made compounds, but these are of natural origin. So the strategy that these kind of the bacteria follow when these kind of antibiotics are given is that enzymatic inactivation of the antimicrobial. So most commonly, aminoglycosides like kanamycin, tobramycin, and amikacin, which we commonly use as the second-line antibiotic in clinical side. So these are very much prone to enzymatic inactivation if the bacterial strain seems to be resistant. So these are inactivated by enzymatic phosphorylation, acetylation, or adenylation reactions. So that is one example for category of antibiotics which are more susceptible to enzymatic inactivation. And the second example is beta-lactams. So these are attacked by beta-lactamase enzymes. So we know what are the beta-lactam drugs, the penicillins, the cephalosporins, and the carbapenems. Okay, so imipenem and all nowadays it is given for treating resistant strains of bacteria. So for that also there could be resistance developed by the bacteria. So here we know that enzymatic hydrolysis by the enzyme beta-lactamase can cause enzymatic inactivation of the beta-lactam drugs. Next slide, please. So here we have an image. This is actually from the World Health Organization, and so they have listed the causes for antibiotic resistance. So we know that inappropriate use of antibiotics is seen globally, whether be it a developed country or a developing nation or a poor income country. So everybody is aware of the fact that antibiotics can be given to treat microbial infections. So, overprescribed antibiotics are possible in some cases, and then patients don't tend to understand the threat that lies behind irregular or incomplete course taken. So, they don't know what is going to come up next, and they don't know the fact that if the same antibiotic is being given to them the next time they have that infection, it's not going to work. It might not work. So, they're not aware of that fact, and hence they tend to use antibiotics inappropriately and inadequate diagnostics. So, when you go to a doctor with a severe infection, the doctor should check as to what bacterial infection or microbial infection you have, and appropriate treatment using proper choice of antibiotics has to be given to the patient. Otherwise, if randomly they tend to give a first line or a second line drug, it just doesn't help them all the time. So sometimes the infection might be moderate. So they might go in for a broad spectrum antibiotic. So in that case also, it is very important that they diagnose and then give the treatment. So at least if it is very essential to start off immediately with an antibiotic therapy, the sample should be taken before administration of the antibiotics to the patient. Only then you will know whether a proper choice of antibiotic has been given or not, or the antibiotic that's being given to the patient will really help the patient or not. So culture can be done, and susceptibility testing can be done, and we can find out whether the antibiotic administered is going to help the patient or not. If not, that can be discontinued once the course is done, and they can be treated with the next choice of appropriate antibiotics. The right choice of antibiotics is very, very important, and for that we need to do adequate diagnosis of the microbial infection. And then selection of resistant microbes in hospitals. So we know that a hospital environment is fully filled with all sorts of microbes. And if you go to tertiary centers, they are very much, the fertile environment for the microbes helps them thrive very well. And if patients are kept in close contact, obviously sick patients have lesser amount of immunity in them, and they tend to take in all the infections that they are exposed to if their immune level is very, very down. So it's very important to control that kind of infections being spread from one patient to another, or even to general public. So selection of resistant microbes is another important cause of antimicrobial resistance. And another thing that we have to follow is proper hygiene. So sanitation and lack of hygiene are major causes. So when poor sanitation and lack of hygiene is there, you don't wash your hands properly after visiting a hospital or touching a patient or handling a sample, or maybe even in your practicals, if you tend to handle the culture of staphylococcus aureus, it is very important for you to wash your hands, clean the surface, your workbench, and keep your safety hood clean, and then leave the lab. So that is how you have to handle microbial infections and these infectious agents. Next slide, please, ma'am. So now superbugs. So have you heard about superbugs? Like while I was an UG student, this it's a colloquial term, but it came into existence in all the news channels and papers because we had a particular strain of E. coli in India, which came up even in international media. It just these news and all, they showed up saying that superbugs. So we refused, saying that no, it's not just our country. We have not generated this particular strain, and later it went into a controversy. So we colloquially call them superbugs. So these superbugs are nothing but bacteria which are resistant to antibiotics. So we can call resistant to multiple antibiotics. So antibiotic resistant threats are seen in specific strains of bacteria. So normal bacteria, like yes, the Enterobacteriaceae families, they tend to have carbapenem resistance. So this particular strain is called carbapenem resistant Enterobacteriaceae. So this is very much common in hospital environments. And the next most common hospital-borne infection is MRSA. So methicillin-resistant Staphylococcus aureus is another important threat. So people are struggling to deal with it, and now we have crossed even that, and it's gone up to vancomycin resistant strains. So methicillin resistant Staphylococcus aureus, they can be treated with vancomycin. Now the bacteria has developed strategies to bring about resistance to even vancomycin. So that could have arisen because of lot of mechanisms that we discussed previously. So the bacteria will adopt a specific mechanism to develop this kind of resistance over time. So the next one is ESBL producing Enterobacteriaceae. So these are extended spectrum beta lactamases producing ones. So that is what ESBL stands for. So these are also seen in clinical samples nowadays. And then another most challenging thing is vancomycin resistant Enterococcus species. So this also is seen in people who are hospitalized for a very long time, and they are into the usage of medical equipments and for a long time. And long time catheterization can also result in enterococcal infection, which could be multi-drug resistant. And apart from that, there are certain specific gram-negative bacteria that I should mention. So Pseudomonas aeruginosa is very important one. So multi-drug resistant Pseudomonas aeruginosa has become very common in the clinical scenario, and multi-drug resistant Acinetobacter, that also has become more common nowadays. And Klebsiella pneumoniae, this particular strain which I have mentioned, ST307, so that was found to be present in a pediatric ward, and it was found to be extensively drug resistant. So that's how they isolated and found this particular strain and how much of drug resistance it could have. So extensively drug resistant strain, this Klebsiella pneumoniae ST307. So just overnight resistance could have not been possible in this much of extensive drug resistant strains, but whereas inappropriate use of antibiotics or incomplete intake of dosages, all these could have led to the emergence of these kinds of resistant strains. Next slide, please, ma'am. So now going into the most important part of the seminar, the actual intention of this particular topic being chosen for the seminar is to create awareness. And student community is a very powerful weapon. And as teachers, we are very much aware that you can carry the message home and into the society also. So you need to know the effect of antimicrobial resistance on public health so that you could realize the importance of creating awareness among public about antimicrobial resistance. So antimicrobial resistance could kill 10 million people every year by projections made by the World Health Organization. So there is an urgent issue which requires a global attention and a coordinated action plan to address the issue. So there are very increasing numbers of drug resistant strains in the community, and it is very much important to make sure that these drug resistant strains do not spread further, and also to stop or prevent the emergence of further resistant strains. So we know that nowadays it's very easy to travel across the globe, and we tend to move very easily from one nation to another, and the frequency of our travel also has increased. So that's why AMR is now a global problem. So a local strain present in a particular community, for example, in a soil, you tend to see a resistant strain in a hospital environment. So that could be carried to any other nation by a person who tends to visit that place and tends to carry that infection to his or her country. So resistant strains are becoming very dominant among the microbial population. So we know survival of the fittest. So that's the strategy, and that's what these bacteria tend to take as an advantage. So the resistant strains, because they have lot of survival criteria and advantages over the susceptible strains, they tend to thrive very well and they become dominant over a period of time among microbial population. So what are the major reasons for this, and what are the outcomes? So major reasons we have seen that improper sanitation, improper hygiene, overuse of antibiotics, inappropriate use of antibiotics, and inappropriate prescription of antibiotics also should be taken into consideration. So the outcomes are like longer hospital stays. So the patient needs to be hospitalized for a longer time. So if the infection is susceptible, they won't even have the need of getting admitted into a hospital. But when it is going to be a resistant infection and there's no help in treating the patient with first line or second line antibiotics, then they have to go for the third line antibiotics, and the patient mostly will be requiring hospitalization. And then the expense of the treatment involved. So hospitalization itself is an additional expense, and on top of that, these third line or higher antibiotics are all very much expensive. And I think one dosage of vancomycin for a day for a patient would cost at least 10,000. So imagine how many families in the general public would be really able to afford this, and are they really responsible for that? Some way or the other, yes. Everybody is responsible for this scenario emerging out. And then even if they are being treated, we cannot assure that there will be complete cure. If in case the higher order antibiotics are also not helpful, and if the bacteria tend to be very much resistant to even higher antibiotics, then the patient is life is lost. Okay, so there is increased rate of mortality that's coming up because of antimicrobial resistance. So these are all the effects of antimicrobial resistance on public health. Next slide, please, ma'am. So now prevention and control. So as an individual, we cannot individually prevent and control this because it has gone beyond that level. So now, as an individual, you can take care of your family. You can be assured that they don't take inappropriate antibiotics. They don't quit any course of antibiotic in between, but you can assure that only within your family. How do you take it up to the society? So this scenario requires coordinated action. So multiple sectors have to be involved in this. So only then we can have better public health outcomes. So it starts with an individual. And then the second category of people are obviously the health professionals, the healthcare providers, the physicians, and other health professionals. And the third category are policymakers, the organizations, the government, and the fourth is agriculture sector. So why do we say agriculture sector? Because antibiotics, antimicrobials, they're also used in producing animals and plants. So there again comes up another challenge. So we tend to consume chicken. Do you know how many antibiotics are used in a farm? They are used inappropriately. So that is another reason. So we need the coordinated action for this kind of prevention and control of antimicrobial resistance. And for that, all these four sectors need to work together. So now coming to the role of individuals. So whenever a health professional, a certified health professional, a physician prescribes you an antibiotic, then only you need to take antibiotics. So even if your physician feels that you don't really need them, you should not ask him or her to prescribe antibiotics, because they know whether it is right or not, whether you would respond or not, and what kind of a microbial infection you have. If at all, if it is a viral infection and you treat them with antibiotics, is that going to help all the time? No. So just you have to follow the prescription of your doctor and make sure that you do not demand antibiotics if they don't advise you to take. And the particular dosage and duration of the antibiotics also needs to be followed very strictly. And you, of course, you have to maintain your personal hygiene by frequent washing of your hands. And while preparing food also, we need to wash the raw materials properly and give adequate time for cooking and then consume the food. So you should always prefer food which are produced without the use of antibiotics. So now the role of policymakers, the organizations and the government. So their role is to make sure that there is an effective national plan in action to fight against this antimicrobial resistance scenario. And then surveillance of antibiotic resistant infections. So continuous surveillance of antibiotic resistant infections is very, very mandatory, and they need to have a record of all the incidences where there's been an outbreak of a resistant strain. So they should make very strong policies and develop programs to prevent and control antimicrobial resistance. And not just making these policies, they also have to make sure that they are implemented. It should reach the public. So it's their responsibility to make sure that the benefits are reached on the other end. So they should also make information on the impact of antimicrobial resistance on human health being available to everybody. So every citizen in a country should be able to have access to the information on the impact of antimicrobial resistance. And now the role of healthcare professionals. So healthcare professionals are people who work in an environment where they deal with patients, microbes, and the different kinds of challenges of resistant strains. So they should not carry the strains from the hospital environment to their home or to the society. So they should ensure that they clean their hands and equipments and keep the hospital environment very clean. And also they should prescribe antibiotics only if required and based on guidelines. So every country has their own guidelines for antibiotic prescription. So they need to stick on to that very strictly. And if there are any antibiotic resistant infections shown up, they need to report it to the surveillance teams of their organization, and also the authorities need to know about it. And the other important role of healthcare professionals is to bring about awareness among patients who are consuming these antibiotics. So they should know if these antibiotics are being misused, what kind of danger they could face. So that awareness they have to create. And also they have to advise patients on what kind of measures they should be taking for preventing these infections. So prevention is always better than cure. And then the role of healthcare sector. So this means the health ministry, the government side. Okay. So the timely measures for control and spread of infections are very, very important. So if the infection has spread to a small group, it is there where you can control the further spread of that infection. If it is not controlled at that point, and if timely measures are not taken, then it might affect a large number of people, and at that point it will be very difficult for management of further spread of the infection. And the second thing is very, very important because our current situation demands this. So the healthcare sector should invest a lot of funds in research and development, of course, which our Indian government is doing. So these should be invested in developing newer antibiotics, vaccines, and also for newer diagnostic tools or techniques. Okay, so a lot of investment in the field of R&D is very important, which the healthcare sector should take care of. And in our country, we have the Indian Council of Medical Research, the ICMR, which takes care of the research R&D development for the medical sector. And then every country has to maintain a national registry for antimicrobial resistance so that they have an account of the outbreaks of resistant strain infections, and also they tend to know like how much effect they have on the community or public health. And then document instances of multi-drug resistant strains, and also the XDR strains, the extremely drug resistant strains. So this is very perfectly maintained for certain infections, like for example, Mycobacterium tuberculosis, which causes tuberculosis infection. That is being very well documented, and to some extent, TB is under control in our country. And then the role of agriculture sector, antibiotics are given to animals, like I told you, in farms they tend to give antibiotics to the animals, but this has to be done only under veterinary supervision. And it's very important for them to prevent the use of antibiotics for the sake of promoting the growth of the animals and preventing diseases. So, well ahead, even before an infection shows up in the farm animals, they tend to use antibiotics so that they think they can prevent the diseases. And also, use of antibiotics sometimes they can promote the growth, because disease prevention is happening, and so the growth of the animals will be very fast. And in the sense, you can avoid diseases. So, loss of animals due to diseases would be the benefit that the farm person could have. And vaccination of animals to minimize the need for antibiotics is very, very important, which of course, if they are running a farm under veterinary supervision, definitely the veterinary person would insist all this. So they should look for alternatives for antibiotics also, so that they can prevent or minimize the use of antibiotics in farm animals. And also safe and good practices should be followed in every step of production and processing of foods. This is not only for animal sources and also for plant sources. And biosecurity is a very important strategy that the farms have to follow. So it's very important also that they maintain proper hygiene at the farms, and animal welfare guidelines need to be followed. And WHO's response to antimicrobial resistance has been very immense. So they came up with a global action plan, and this happened in May 2015. So that's called the GAP, so Global Action Plan on Antimicrobial Resistance. So they had 5 strategies. One was to improve the awareness and to understand what is AMR, and then for surveillance and research, and then to reduce the infections, and to optimize the use of antimicrobials to channelize that, and also to make sure that the investment is happening in countering antimicrobial resistance. So we know that a week is being specially dedicated, and that's called World Antimicrobial Awareness Week. So this has been happening since 2015. So in tertiary care centers, you can see them doing this. Like every year between 18th to 19th, 18th to 24th of November, they tend to do this awareness week. So this is being followed worldwide. So just that they have to encourage good practices among the public and the healthcare workers and policymakers in order to avoid emergence of resistant strains and to control infections due to resistant strains. And antibiotics handled with care was the previous slogan that they had, but now it is antimicrobials handled with care. So antibiotics, it refers only to the agents that are used to combat bacterial infections, whereas antimicrobials are those which could be used for viral, fungal, and malarial parasites also. So other measures by the WHO are global antimicrobial resistance surveillance system. So their job is to collect, analyze, and share the data of AMR on their WHO website. So this is very important for decision making. So be it local or global action, they can make the decisions based upon this data. The second thing is global antibiotic research and development partnership. So this partnership is with the Drugs for Neglected Diseases Initiative, so DNDI. So along with them, WHO has come up with this plan. So research and development, that is R&D through public-private partnerships. So because of that specific kind of public and private partnerships, lot of funds can flow in, and lot of R&D activities can take place. And this would lead to the discovery of newer antibiotics, newer vaccines, and newer diagnostic techniques or tools. And the last one is interagency coordination group on antimicrobial resistance. This again is to coordinate between different nations along with the WHO so that they can come up with decision making policies. And here ends the seminar. And thank you so much for this opportunity. I believe that this seminar would have imparted thoughts in you to spread the awareness on antimicrobial resistance and to keep our society safe and very much protected from resistant strains of microbes. Thank you.
Thank you, ma'am, for your valuable information. So, as ma'am said, let's all prevent antimicrobial resistance together. Now, I request our beloved head of the department to address the gathering. Over to you, ma'am.
Good morning to everyone present in this forum. Hope you can hear me. So it was a nice presentation, crystal clear presentation that would enable students a better understanding view on what is antimicrobial resistance. And thank you, Dr. Udya Lavanya, for your presence in spite of your busy schedule today with us. And
Thank
I
you,
would
ma'am.
like to see many more such presentation in the future as well. So it's been a long way for the Department of Microbiology to connect online after this COVID. Right? When we started with what is called as COVID, when we were in a lockdown period, virtual seminar were the only forum where we were able to interact with the children online. So that is where brought the educational forum together. So again, we would like every week to have such programs wherein we can see resource people interact with the children to make them understand what microbial creatures are all about to them. So henceforth, we will be continually doing such YouTube live streaming for the betterment of the society as well, for the children as well to know what is this all about. And thank you for your generous availability in spite of your busy schedule today with the children. And if there are any more doubts the children has, they can maybe comment in the box the question so that it can be put forth by the host to the resource speaker. Thank you, and let us all be blessed by God. Thank you.
Thank you, ma'am.
The session is now open for discussion. Students, if you have any doubts, you can just type in the comment box.
Maybe Kalpana, if there are no questions from the students, we can wind up the session.
Yes, ma'am.
Yeah.
Now I would like to call upon Ms. Munasira Behim, Assistant Professor from the Department of Microbiology, to convey vote of thanks.
Good afternoon, everyone. The best and beautiful things in the world cannot be seen or even touched. They must be felt by heart. Thank you is one such prayer among them. I consider it a great privilege to propose a vote of thanks to everyone who have witnessed it as a memorable and successful webinar. First of all, I would like to propose a hearty vote of thanks to our resource person, Dr. Udaya Lavanya, for gracing today's webinar. Thank you, ma'am, for making excellent presentation and making this webinar interesting and meaningful. I extend my gratitude to our beloved HOD, Ms. Gauri ma'am, for constant help and guidance. My heartfelt thanks to our department faculties for their contribution in all our efforts. I also thank all the students for their enthusiastic participation. Have a nice day, everyone. Thank you once again.
Thank you, ma'am. Thank you, one and all.